Alrighty. So today is something very interesting. We're going to be talking about microbial genetics. We're going to be talking about microbial genetics. And we're going to see that genetics, for you guys took anatomy and physiology, you know about human genetics. Well, today we're going to learn about the genetics of the unicellular organisms, about the microbiology genetics. We're going to see that some things are similar and other things are different between us and the microbial genetics. You need to remember some basic things that have to be set in stone. Number one, what is genetics? Genetics is the study of hereditary transfer of information from one generation to the next. That's part of reproduction. We're not only concerned with the homeostasis of the organism, we're also concerned with the homeostasis of the entire population. So we have to be very careful how to define genetics, because what's the ultimate goal of genetics? Is the reproduction and the preservation of the entire human species for us, and for microbes, they want to stay around too. Microbes they don't want to extend. Only dinosaurs have extended because they have failed to propagate the because they have failed to propagate their genetic information to the next generation. So, what is genetics? Transmission of information from one generation to the next. And what are we transferring exactly? We're transferring genetic material, meaning the capacity to make proteins. I really need you to understand this. If you took microbiology or if you took biochemistry with me, you see my favorite chart is this, that we have DNA, and the DNA is going to be transcribed, or let's call the process transcription. You're going to undergo transcription, and the transcription is going to give us RNA, and the RNA is going to be undergoing translation. Where does RNA translation take place? In the ribosome. And where does transcription of the DNA take place? In the someone saying ribosome?
No, I said ribosome for RNA.
Okay, very good. And ultimately, we get proteins. So proteins are what make the cells alive, because proteins, you have proteins that are structural proteins. We have structural proteins, and we have functional proteins, structural and functional proteins, and that is to preserve the structure and the function of the cell. Okay, and we said that we have 3 types of RNA. What are the 3 different types of RNA?
tRNA.
Excellent. tRNA. I wish that they called it translation RNA, because that's what it does. But what does the tRNA stand for? Listen to the question. tRNA. We already finished transcription. So now we're moving beyond transcription. There is no way RNA should be called transcription RNA, because it has nothing to do with transcription. Transcription is when we transcribe the DNA. Everybody listen. Look up here. DNA is the doctor. You go to the doctor's office. The doctor will give you a prescription. You take the prescription to the pharmacy. The pharmacy will give you the medication. You cannot jump and go to the pharmacy and say, can you give me antibiotics? You have to go to the doctor. So here, the doctor is the DNA. You go to the DNA. The DNA is made up of chromosomes. Chromosomes are divided into different segments. Gene 1, gene 2, each segment. A segment is like an aisle. An aisle where you find, but remember, each gene will code for one protein only. So there is hemoglobin gene, there is keratin gene, there is collagen gene. So they have different types of genes to give us the target protein. So we're going to go to the doctor, to the DNA. We're going to transcribe the DNA. Then we're going to get the RNA. We're going to pick up that purple prescription. This is the RNA. And then we're going to go to the ribosome or pharmacy. The pharmacist, the ribosome, is going to scan and read that prescription, the RNA, and read it to give us our desired product, the medication that we want, the protein. So we have tRNA and we have rRNA and we have mRNA. So tRNA stands for look before you answer. Don't shout an answer. Look, take a while, listen, and think, and give an answer that you have already thought about. Don't just yell something. Yes, please. Transfer RNA. Very good. Because it participates in the transfer of amino acids to the polypeptide, the growing polypeptide chain in that P position of the ribosome. But I wish they called it translation RNA, but they call it transcription RNA. So we have tRNA, we have rRNA, and we have mRNA, the messenger RNA. That is for translation. Translation. So we call it transfer. Transfer RNA. If I gave you a question and told you what does tRNA stand for, translation or transfer, absolutely you cannot choose translation. It's transfer. It's transfer RNA, but it's transferring during the translation. rRNA is ribosomal RNA. And this is the one that makes your ribosome. So the ribosome has multiple subunits, and each subunit is made up of the rRNA. This pharmacy is made up of bricks of rRNA. So the mRNA is being translated by the two huge rRNAs. Is that clear? I told you the rRNA is so important that it has its own special headquarters. It has its own special, if this is the nucleus, there is a little bubble inside the nucleus where all the rRNA is stored. What do we call that little place? Raise your hand. Raise your hand. What do you call this little? Yes, Devon. I cannot hear you. Yeah, excellent. This is the nucleolus. Excellent, excellent, excellent. Then we have the mRNA, and this is the actual purple, that's the actual purple prescription. The two RNAs are helping translation. tRNA helps the transfer of amino acids, the growing polypeptide chain. Ribosomal RNA makes the ribosome, and and the messenger RNA is what we actually read. What we actually read to get the target protein. Then we get our protein of interest. When you look at sickle cell anemia, your natural red blood cell is supposed to look like that. It's supposed to look like a disc, called discoid. It has to be discoid red blood cell, and it has to be double depressed like that. It's called bi. Bi means two. So biconcave, meaning that it's concave from two different sides. This is the most thermodynamically favorable structure for the red blood cells to maneuver. inside the blood vessels and to be able to carry the oxygen. That's the best shape. Now, people who have sickle cell anemia, their red blood cells are not biconcave. They are not discoid. They are sickle. Sickle. They're sickle cells. Why is that? Because in order to be discoid, in order to be biconcave, you have to have proteins that set you up in this type of staple you and hook you up in a way that makes you discoid, in a way that makes you discoid. See these proteins? These are known as cytoskeletal, like skeleton. Cyto means cell. Remember cytology? Study of the cell, knowledge of the cell. Cytoskeleton, that means the skeleton of the cell, like the scaffold, like construction. So there's proteins that hold the cell. Those are structural proteins, okay? Those structural proteins in people who have sickle cell anemia, remember, they are structural proteins, so they're here. They're structural proteins. Those structural proteins, you're never going to get them. You will never get those structural proteins unless you go down this route, right? If you want to become a nurse, you got to go to nursing school. You're never going to become a nurse if you don't go to nursing school. Same thing. You're never going to get protein unless you go through this stepwise process. 1, 2, 3. This is the most important thing in biology. We call that the central what? Dogma, the central dogma of biology. The central dogma of You sure? You sure? You doubt. Some people in this life, their own name. What's your name? Are you sure? Yes, it's the central dogma of biology, the central creed of biology, because biology is based on proteins that run life based on the transcription and the translation of the transcription of DNA, translation of RNA. So for people who have sickle cell anemia, those proteins here, the construction proteins that make you, twist you, bend you, can we have them? No. Why? Because there is the gene here is supposed to have GLU, glutamic acid. GLU stands for glutamic acid. Who belong in the gene? Glutamic acid. But due to a mutation, no glutamic acid is going to become, what is it going to become? Valine. And that's the problem. Who do we need to have to become discoid and biconcave?
Protein.
You need to have the protein, but what is the amino acid that should have, the gene we're trying to transcribe, what amino acid is missing in there? Glutamic acid. And who did we end up with? Valine. So this is the central dogma of biology. Is it only for human? No. For animals? For even bacteria. Everybody, no matter, as long as you're in life, as long as you are in this God earth, you're never making proteins unless you go through the central dogma. That's why we call it the central dogma of biology. That's why when we look at the bacteria are not complex. They don't have organelle. They don't have the cytoplasm that we have. They don't have mitochondria. They don't have a lot of organelles, but you can bet that they will absolutely have what? They will absolutely have DNA. They don't even have a nucleus. Does bacteria have nucleus? Bacteria has no nucleus, but they have a DNA, because if they have no DNA, they have no genes. If they have no genes, they're not going to be able to to prescribe RNA. And in addition to they gotta have DNA, what else do they have to have? Ribosomes. Excellent. They have to have ribosomes. But is their ribosome gonna look like our ribosome? Was your high school cafeteria the same like your college cafeteria? So it's different. Yeah, that high school cafeteria was disgusting.
This one is good.
Yeah, there's no cafeteria I talk about. I'm talking about like about the big colleges. There's a cafeteria here that I don't know about. I'm going there immediately at 6 p.m. Anyways, so that shows you the level of the structure and the function of the genome. The level of the structure and the function of the genome. You see, if you look here, that's what I was talking about. If you look into my mouse, do you guys see the mouse? You do not see the mouse. Okay. This here. You transcribe the DNA and then you translate it, and you see it's amazing. What is this showing you? This microorganism, a parasite, we took one cell from it. It doesn't have a nucleus, but it will absolutely have what? DNA. Maybe they don't have a nucleus with as much genes like we do, but they gotta have DNA. Some of them have circular plasmid DNA. Some of them have like fragments DNA. Even a virus has DNA. DNA is like the soul of the organism. If you have no DNA, it means you cannot transcribe and you cannot translate the proteins. Okay. The genome is the collective genome, the collective set of genes that are available in your in your nucleus. So the sum of the total of the genetic material, DNA in the cell, and it exists as chromosomes. So you have to remember that this DNA, if you spread it out, spread out the DNA, it'll be as big as a football field. Did you guys see the Super Bowl? These people have nothing to do. They are not professors. They don't have to grade no exam. They have nothing to do. But anyways, the DNA is able to wrap that Super Bowl stadium multiple times. So how does it fit inside the nucleus? Because the way, sometimes I wanted to pack up, like I was traveling, and my backpack, my clothes couldn't fit in my backpack. Then my mom came in a way, and she folded them, and everything fit. That's exactly what happens. My mom and my sister, I'm sorry. Anyways, so what happened was we looked at what happened in the DNA. The DNA is coiled. The DNA is wrapped around proteinaceous structures into coils. What do we call those proteins that we hook the DNA around? This is bad. I'm sorry, histones. Yeah, the DNA is called, the protein is called histone, and the DNA. But when they are bound together, when the histone is alone, it's just protein, it's called a histone. But when the DNA is wrapped around the histone, that collective structure is called what? So we have a helical coil, but when you look at DNA we look at a coil of histone with DNA around it. What do we call that? What's the name of that? Yes, please. I cannot hear you. I wish I can hear that part. I wish. DNA gyrase is the protein that wraps, the enzyme that wraps it, but it's not the name of the structure. It's the nucleosome. The nucleosome. The nucleosome is the structure of the histone, and the DNA is wrapped around it. So some appear in non-chromosomal sites. That's what I was telling you. We're not the only ones that have DNA. Plants have DNA, but they don't have it in no nucleus. They have it in the chloroplast. The mitochondria has a DNA. The mitochondria, please don't forget, you know how everybody knows in high school that the mitochondria looks like that. It was my favorite organelle growing up in high school. I couldn't wait to draw the mitochondria. I thought it was cool. But this is incomplete because something is missing. What is missing? No, the inner membrane is right here. The inner membrane is right here. Please raise your hand so I can hear you. If it's true, it's true. If it's false, I correct it. Yes.
I get that two chambers.
Two
membrane.
Two membrane. It's right here. That's the outer. That's the inner. You don't see two membranes. That's the outer. That's the inner. That's two membrane. Something else is missing. Yeah.
That bar shape.
Sorry. Not the ribosome. Why would the organelle have an organelle inside it? No, the ribosome is to make proteins. Bacteria has a ribosome. The whole cell has a ribosome. The human cell has a ribosome. Why would an organelle like the mitochondria have another ribosome inside it? The mitochondria is to make this ATP. The mitochondria is to do apoptosis, program cell death. Yes. So the cristae is here. It's the circular, look there. Some appear in non-chromosomal site. So there is a circle in here. This circle is called a plasmid. Plasmid DNA. It's a circular plasmid DNA. So now the picture is complete.
The plasmid don't show in it doesn't show in the mitochondria in the cell. It's not showing.
It's showing.
But
That's why we drew it in there.
But we can't see it though.
Of course you can because you need the microscope, but it's there. And this circular DNA was a discussion among the scientists. Why the mitochondria has DNA? Is it a cell? Is the mitochondria a cell? No, it's an organelle. Why you gotta have your DNA? DNA don't belong there. DNA belong inside the nucleus. Ribosome has no DNA. Endoplasmic reticulum has no DNA. The purebred Golgi body has no DNA. Lysosome has no DNA. Why you mitochondria gotta have DNA? Then they came up with something known as the endosymbiotic. The endosymbiotic theory. What does the endosymbiotic theory mean? If you look at the words endo and symbiotic, I was telling you in class, endergonic, energy is being stored. Exergonic, energy is exiting. So endosymbiotic, yeah. Endosymbiotic means something inside. Good, so you got that part. Endo means it's inside. What is symbiotic? Raise your hand. Yeah. Yes.
Something is working together.
Absolutely. So that's the theory of this. Yeah, endergonic and exergonic, that's for energy. If we want to save energy, we do anabolism, endergonic. We want to get energy out, we do exergonic, catabolism. Endosymbiotic means now they believe that this mitochondria used to be a prokaryote cell. Because prokaryote cells have a circular DNA. The bacteria has, who has circular DNA? Bacteria. We have X sister chromatids. How does our DNA look? Our DNA looks like X's. What does the bacteria DNA look like? Circles. Now the scientists say, so that means maybe millions of years ago, there was a bacteria who swallowed another bacteria. or another eukaryotic cell, and you end up with this mitochondria with the retained DNA. Are we clear? Okay, now everybody got to have DNA, except viruses are special. Viruses, we talked about that last week. Viruses have DNA, so they have vDNA. What is vDNA? vDNA is viral DNA. Then they transcribe it and get viral RNA, and then they translate it and get viral protein. And then they kill us. Some viruses have viral DNA. Some viruses have RNA. So let's look at the complex again. You are a human. Let's say, God forbid, you are a human. Human, you have human DNA, human RNA, human proteins. And then, God forbid, you had a HIV virus. So now, what does the HIV virus have? The HIV virus has excellent, vRNA. So what does the virus do? So it has its vRNA, and it doesn't have ribosome. It needs to get V proteins. But if you take the viral RNA and you give it to the ribosome will say, nope, this is not the purple transcript. We read it. So now the virus has an enzyme called reverse transcriptase. Reverse transcriptase. Meaning that this enzyme here, reverse transcriptase, is going to reversely transcribe viral RNA to give us viral DNA. Excellent. So that's why they call it reverse, because it works in the opposite direction. You see, in the central dogma of biology, we go down this direction, from DNA to RNA to protein. But the virus has viral RNA, then it has a reverse transcriptase to go back to give us viral DNA, and then sandwich it in the middle of your human DNA. Now, when your own cell is trying to do transcription, is it going to find out that this is viral DNA? It will never. So it's going to, your own human transcription RNA polymerase is going to give you viral RNA. And your own human ribosome is going to translate the viral RNA to give you viral proteins. So that's how the rRNA viruses work. And this is the AIDS, the HIV. It's not as bad as it used to. Back in the day, AIDS used to be the number 4 killer of people worldwide and in the United States, especially in economically disadvantaged, medically underrepresented communities. It wiped out the people who make below like $50,000. It was crazy how this disease was just killing the poor people, the indigenous people, the people who are suffering. And then obviously everybody had encountered AIDS at that time in the 90s, in the early 90s. But who suffered the most from it? The affluent people, they didn't suffer from it that much. Okay, so now it is the number 20 killer. It used to be the number 4 killer. Now it's the number 20.
What's the number 1?
The number 1 killer is? No, number 1 killer is infectious diseases, because you have to understand the whole world is not like the US. You're living in the most advanced, technologically most advanced country in the world. So to us, it's cancer. That's the number 1 killer. But the rest of the world is suffering with so much infection microbes. That's why most of the people who die outside like for example, in Africa, Latin America, and Eastern Asia, The most amount of people, they don't die from cancer because cancer is even lifestyle. The choice you make, the types of food you eat, the sugar you eat, the chicken, that's why I told you guys earlier in this Ramadan, we're taking our health back. I'm taking back my health. We're not eating a lot. We're not eating like drinking soda and all that kind of stuff. So sometimes your wealthy lifestyle leads to cancer. Then have a wealthy lifestyle. And we gotta learn today in another other lecture. It's expensive to have a clean world because those bacteria require a lot of different types of antiseptic techniques to make sure that we don't not live in a contaminated world. And unfortunately, they can't afford that. They can't afford that in Africa. They can't afford that in Latin America, in the Caribbean, in Eastern America, in Eastern Asia.
They start injecting those animals with stuff to make them grow faster, then people will not be able to catch cancer.
Yeah, but they don't think human. They think for money. They don't think about what's the best interest of the people. They think about what makes more money. That's all they care about. Okay, all right. So we talked about that. Chromosome is a discrete cellular. The chromosome is a discrete cellular chromosome is a discrete cellular structure composed of a neatly packaged DNA molecule. So remember, we have eukaryotic chromosomes located in the nucleus, are multiple and linear, but wrapped around a coil known as the nucleosome. Bacteria chromosomes, look there. They are single and circular. So the chromosome, so that's the nucleus. These are the chromosomes. And that's the nucleolus inside it. This is the mitochondria. And you see that this is called, what is extra chromosomal DNA? What is extra? Look at the name. Always look at the name, will give you a hint. Extra chromosomal DNA. What does that mean? It means that it's like the one in the DNA, that plasmid, because it's extra outside from the nucleus. Okay, genotypes and phenotypes. Genotypes is the alleles, homozygous, dominant, or heterozygous, or homozygous, recessive. That's your genotype. When the genotype manifests physical features, are you tall? Are you short? Or do you have what color your eyes? That's the physical phenotype. So one genotype gives you the phenotype. Okay, so the DNA molecule, that's what I was talking about earlier. Everybody look at where my mouse is. You see where my mouse is? Now, this is the chromosome. If you look at the chromosome, it's multiple coil structures, and the more you loosen up, you loosen up this coiled, condensed chromosome. You keep loosening it up. What do you see? You see those balls? You see those circles? What are those? Those are the nucleosome. You see where my mouse is? Can you see where my mouse is? Okay. I can't even see where my mouse is. I need my glasses. So the DNA wrapped with histones around it. And then, so what happens if we need to make transcription? We're going to uncoil it. This DNA is not going to stay like that. That's why I said the DNA process has a lot of transcription factors and enzymes that loosen up, relax DNA to perform transcription. Okay. The DNA, we know that the DNA is a double helix, is made up of a nucleotide. Everybody listen. What is a nucleotide made up of? Nucleotide has 3 structures. What are they? Yes.
One phosphate, one nitrogenate, and one sugar.
Round of applause for Tiffany. You got 1 extra point on the next exam. That was amazing. And that was like shocking. Yeah, like she said it, she was shocked. She was so shocked she couldn't say that word. Night, but she said it. She came through. Very good. She's a strong nurse. Now I have another tricky question for extra credit too. Who's up for it? One more question. So Tiffany said, if you look up there, look there in the monitor. We have a deoxyribose sugar, meaning a sugar, because in the RNA we have a sugar also. So we have a three-headed beast. A sugar, a phosphate, and the nitrogenous base is going to give you a nucleotide. One of those, if I strip them away, I'm going to turn into something else. What is it and what do I become? Raise your hand, because if somebody hears it and says that they're going to get the extra credit. Those three, if I subtract one of them and remain with the other two, I'm going to turn into something else. What is it? Yes.
I'm not sure. Okay. I think it's the sugar. I mean, the phosphate. Okay.
So the deoxyribose and the sugar,
Yeah,
deoxyribose and the phosphate together. Okay, who else want to try? Yes, Bacola. I cannot hear what you said. Do I get ATP?
If you strip away,
So how are you going to get ATP if you strip away the nitrogenous base? And ATP is adenine triphosphate. Adenine is a nitrogenous base. Yes. I cannot hear you. Somebody says what you said. You need a microphone. I'm gonna bring that little microphone. Yes, please. Absolutely. Round of applause for you, Gwen. Good job. You remove the phosphate. You don't keep the phosphate, because if you keep the phosphate with the sugar, you have a phospho sugar. What is that? But when you remove the phosphate, you have a nitrogenous sugar. That's a nucleoside. You add a phosphate to it, it becomes a nucleotide. Again, sugar, nitrogenous base, sugar, and phosphate. Those are the three that will give us what? Those three will give us what? Nucleotide. But if you only, let's see if you have 1, 2, and 3. But let's say I chop off the phosphate. No phosphate. Phosphate gone. Now you have nitrogenous base and a sugar. What is that now called? Nucleoside. Nucleoside. Nitrogenous base covalently bond to the alpha carbon. So we have adenine and thymine. They are 2 hydrogen bonds each. Guanine and cytosine, they have 3 hydrogen bonds each. Do you see where my mouse is? Adenine and thymine have how many bonds? 1, 2. Guanine and cytosine have how many bonds? 1, 2, 3. Clear? Are we clear? Antiparallel arrangement. So in one strand, the helix runs in a 5' to 3'. The direction in the other side is oriented from 3' to 5'. 3' to 5'. That's why they're anti-parallel. Can you tell an anti-parallel? Look in there. Red is 5', green is 3'. Look at the opposite strand. Who's facing the 5' end? Is it another 5' or the opposite 3'? That's why we call it anti-parallel. Anti-parallel. Who can answer this question? In DNA, Adenine is the complementary base for, raise your hand. Yes, please. Can you say it? I did not hear you. Adenine is the complementary base for thymine, and cytosine is the complementary base for guanine. Very good, very good, very good. Keep it up, Christy. You're gonna get extra credit. Just keep it up. Give the participation. You don't want extra credit? Replication occurs on both strands simultaneously. We said that the DNA, what's DNA replication? Replication is, let's say you have one pharmacy. You want to open up pharmacy number 2. Are you going to cut the medication in half and send it there? Or are you going to double the medication that you have and send it? You're going to double it. So the same thing with the DNA. You're going to divide. All cells going to divide. Bacteria cells, they need to reproduce. They need to develop. So they have to divide. Human cells, eukaryotic cells divide, and bacterial cells divide. But the process of the division requires the duplication of the DNA. But when we duplicate the DNA, the 2 strands are separated, exposing the nucleotide sequence to serve the templates. Now, let's look at the overall replication process. Looking at the overall replication process. If I tell you describe replication in 2 words, semi-conservative and discontinuous. DNA replication is semi-conservative and discontinuous. DNA replication is semi-conservative and discontinuous. Why is that? That's number 1. That's number 2. You should be able to explain why is DNA semi-conservative and why is DNA this replication. What is semi-conservative and discontinuous? It is the replication of the DNA. Replication of the DNA is semi-conservative and discontinuous. Yeah. Excellent. And then which one is it? 1 or 2? 1. Everybody look there. Everybody look there. So if you look now, the DNA is blue, right? Blue. Mm-hmm. DNA is blue color. So we unzipped it, we chopped it in half, we opened it, and when we opened it, we did transcription, we made the purple copies. So what do you end up with? You end up with the original blue strand, that's called the parental. That's the old original strand. The parental strand, blue, and the daughter strand, the one that you made a copy. What do we call that? Daughter strand. So you began with a parent, you ended up with a daughter. That's why we call it semi-conservative. Discontinuous. We talked about how the DNA is anti-parallel. 5 prime, 3 prime, 3 prime, and 5 prime. The replication process is always from 5 prime to 3 prime. So the DNA polymerase works from 5 prime to 3 prime, but because the other strand is inverted upside down, is not gonna, on the upside down one, is not gonna be able to replicate in a continuous way. It's gonna stop and go. It's gonna lag. That's why we call it discontinuous. The overall replication process. You have the DNA replication. And what happens in the replication? We began with blue. What did we end up with? We ended up with 2 cells that each has an original blue and the second one has a purple strand, meaning one has a parental strand and the other has a daughter strand. Okay, the replication of the DNA starts, so the point at which you start the replication is called the replication fork. The replication fork. The DNA replication is semi-conservative, meaning that each of the two molecules created, they're gonna contain an original strand and a daughter strand. Why must the lagging strand of the DNA be replicated in short pieces? Is it A, because of limited space, or is it B, to avoid distortion of the helix, or C, the DNA polymerase works in only one direction, or to make proofreading of the code easier? C. I just said it works in one direction. One is upside down, so it's going to be cutting, chopping up. It's going to be working backward. Isn't that what we talked about earlier? Let's look at what we have here. That's the DNA of the bacteria. Then what do? We transcribe the DNA. What do we get? Look at where everybody, look where my mouse is. That's the DNA, and we transcribe the DNA. We get tRNA, we get mRNA, and we get rRNA, that's transcription. Then we translate in the ribosome, and we get our protein. What do we call all this process? What is it called? Central dogma of biology. Very good. Okay. Okay, now we look at the gene-protein connection. So there are groups of 3 consecutive bases. Those are known as triplets. So each DNA, everybody look here. You see this purple? That's your DNA. When you go from DNA to mRNA, this arrow here, what do we call that process? Transcription. So we transcribe based on 3 triplets of nitrogenous bases in order to get mRNA. And then the mRNA, when you go from mRNA to amino acids, what do you get? From mRNA to amino acids, what do you get? Amino acids. From mRNA to amino acids, that's the process known as translation. So how do we translate? Every 3 codons give you 1 amino acid. Remember, when I was talking about sickle cell anemia, one of those, look where my mouse is. So for example, CUG is supposed to give you leucine. For glutamic acid, 1 is going to be mistakenly, so for example, we get the glutamic acid with UGA. UGA gives us glutamic acid. There was a mistake, so now you no longer have UGA, you have UGC. So instead of ending up with glutamic acid, what do you end up with? You end up with valine, and that's why you can't have the proteins who are going to hold the structure of the cell. Major participants in the transcription and the translation. So you must remember that there is 1 nitrogenous base you find in the DNA, but you will never find it in the RNA, and some nitrogenous base you find in the RNA, but you will never find in the DNA. What is that? Thank you. Very good. Good. Okay, major types of RNA. We talked about the messenger RNA is a sequence of amino acids. In the protein, it carries the DNA, and it gets translated. But not all RNA gets translated. Messenger RNA gets translated, but transfer RNA does not get translated. It just is a transfer. It participates in the transfer process during the translation of mRNA into DNA. Ribosomal RNA, that's the large structural RNA that makes the ribosome. Primer RNA, so you have an original, that's an RNA that can begin a DNA replication, primes DNA. Are we clear? So the ribosomal RNA, so you have the prokaryotic 70S ribosome is a particle composed of tightly packaged ribosomal RNA. So now if you look, that is the RNA that makes for us the ribosome of the bacteria. The ribosome of the eukaryotic cell, does it look like the ribosome of the bacteria? It does not. Transcription, that's the first stage of genetic expression. So during transcription, we have 3 stages. Initiation is when you hook up the mRNA transcript to the ribosome. Now you initiated the process. So now the ribosome is biting with its teeth on the mRNA. What's the name of the location at which so that's the transcription. We can now begin transcribing the bacterial DNA. Okay, everybody needs to understand this. This is really, really, really important. You have to write this down, all of this. You have DNA that gets translated to give us RNA, and RNA is translated to give you proteins. What's the name of the enzyme that performs this transcription? There's an enzyme. There's an enzyme that does this job. Transcription is a process. Who does this transcription process? Is an enzyme known as? No, not transcriptase. It's called ACE. And it makes RNA. Is RNA a polymer or a monomer? Is a polymer. So it makes polymers. That's why we call it polymerase. Are we done? No? What kind of polymer does it make? Does it make an RNA polymer or does it make a DNA polymer? So what's the name of the enzyme? RNA polymerase. So who carries out the process of transcription is RNA polymerase. Now, RNA polymerase, we call it, what did it depend on? What did this RNA polymerase depend on to make RNA? What did it read? DNA. So we call it DNA dependent RNA polymerase. DNA dependent RNA polymerase. Now, if we try to do replication is this. This is replication. When you make 2x, 2 times, you double up the DNA. You're trying to open another pharmacy, you double up all the medication, then you carry it over to the other pharmacy. Replication is when you double up the DNA. Who does that? So somebody got to do that job. Who does it? It's an enzyme. What's the name of the enzyme that does replication? Stop saying transcriptase. That's not the name of the enzyme. That was just specific for the HIV virus. But we call it DNA-dependent RNA polymerase if it makes RNA. Now help me make the name of the enzyme that makes DNA from DNA. So that's obviously going to be an ACE. That's an enzyme, ACE. And it makes, is DNA a polymer or a monomer? Ribonucleic acid. deoxyribonucleic acid. Both of them are nucleic acids. Both are at the top. When we drew that arrow thing from nucleotide, nucleic acid were at the top, so they're polymers. So DNA is a polymerase. Polymerase. And it made DNA. Did it give us RNA or did it give us DNA? DNA. So that makes it a DNA polymerase. What did it depend on in order to give us the DNA that we wanted, the 2 times, the 2x? What did it? DNA. So that's why we call it DNA dependent DNA polymerase. So you see, for transcription, we use DNA dependent RNA polymerase. For replication, we use DNA dependent DNA polymerase. Now let's go to our HIV virus. I really hope you guys can get this one. So this HIV virus goes this reverse transcriptase. So reverse transcriptase, reverse transcriptase. Reverse transcriptase is obviously an ACE. Help me. We're going from here to here. Okay, so help me. Is an enzyme. What kind of enzyme is an ACE? Is an ACE that makes polymers or monomers? I don't know why you guys keep saying that DNA is a monomer. DNA is deoxyribonucleic acid. Is a nucleic acid. Nucleotides are monomers. Nucleic acids are polymers. So reverse transcriptase is going to give me DNA, so that makes it a polymerase of DNA. Is the name done? No. What did it depend on? What did it depend on to give me the DNA? RNA. Because it has RNA, it read RNA and reversely transcribed DNA, so we call it RNA dependent DNA polymerase. You get it? Clear? Are we clear?
About the biology. Biochemistry, another question.
Yeah, because it's all the same. Microbiology, anatomy and physiology, biochemistry, we're all studying life. Pharmacology, you will see it again. So that's why we're going to look at medications that destroy this. If you want to kill a bacteria, if you want to cure the HIV epidemic, you have to stop the reverse transcriptase. Okay, so those are the polymerases. We talked about that. Translation, the genetic code. Okay, so that's the translation. Okay, you have to understand that the DNA is absolutely never the RNA is never translated. The RNA is never translated as one ribosome. There is multiple ribosomes. So we call it the polyribosome. The polyribosome. Eukaryotic transcription. Eukaryotic transcription. Okay. So eukaryotic transcription, it's very interesting that the microbes, the bacteria, they have transcription and translation happening at the same time. That's something we can do. They're crazy. They do things that way. So microbes, bacteria, they're able to transcribe and translate at the same time. Okay, when we look at the regulation of the protein synthesis, there are operons. Operons, these are regions in the DNA. These are genes that are regulated to be active only when their products are required. There are two types of operons. Sorry. There are two types of operons. There is the reversible. Those are genes in a series that are turned off by the product synthesized. Okay, when we look at the lactoperon, the lactose operon, it is made of three segments. There is a regulator, that's the gene that codes for the repressor and has a control locus. It's composed of a promoter and an operator and has a structural locus. It has a structural locus. It is normally off, but in the absence of lactose, the operator, the repressor is going to bind with the operator locus and is going to block the transcription of the downstream structural processes. Okay, the arginine operon is repressible. It is normally on and will be turned off when the product of the pathway is no longer required. So when the operon is on, the arginine is being used by the cell, and an repressible operon is going to remain on when its nutrient products, you see like amino acids and arginine, they are in great demand by the cell. Okay. Oral mutations. We discussed this very crucial concept, and the perfect example for it is the sickle cell anemia. Sickle cell anemia looks like it's a problem with the amino acid. We needed to have a glutamic acid. We ended up with valine. That's what it looks like. But that's not actually the actual problem. What is the underlying problem that caused that? What caused sickle cell anemia? What caused sickle cell anemia? Raise your hand. Somebody. Extra credit. Yes, you got a hand up. The veins?
The valine.
Valine. So what happened? One word answer. Absolutely. So there was a mutation in the gene. The problem is not down there. The problem is up in the nucleus. There is a problem in the gene, so we didn't transcribe the proper codon. So now there are different types of mutations. There are spontaneous mutations, and there are induced mutations. So spontaneous mutations, they have a random change in the DNA due to errors in the replication that occur without a known cause. Induced mutations result from exposure to known mutagens, physical primary radiation, or chemical agents that interact with the DNA in a disruptive manner. Now, those are the different types of mutations. But in the end of the day, they're all mutations, meaning we don't have an OK gene. It's like the doctor writes something wrong. Like you go to the doctor for antibiotic and they write for you something for a skin infection. So you're gonna get something that you cannot really make any use of, like the protein that's supposed to hold the red blood cell in a biconcave discoid shape, but they didn't. Now let's examine the different types of mutation. The first one is called a point mutation. Point mutation means at a point of a codon. Point meaning it's involving one codon. So we add by mistake, or we remove by mistake, or we exchange a codon. So for example, let's say your phone number is 1234567. If I remove one, it's going to be 2345667. I can't call you. If I switch 1 and 2, so now instead of having 1234567, I have 2235 is not you. Or I can have an addition. Or you have 1234567, and I add one more number, it's not going to dial. For example, that's a point mutation. There's another mutation known as missense mutation. Missense mutation. Missense mutation causes a change in a single amino acid. Missense mutation causes a change in a single amino acid. So that means the overall mutation leads to a different amino acid. That sounds like sickle cell anemia because it causes a change in a single amino acid. So it's crazy how only one amino acid messes up the whole red blood cell. So that's called missense. Nonsense is that when you have a normal codon, you know when the ribosome is translating, it starts with a start codon. Who remembers what's the start codon? No, that's the stop codon. You stop at that. I asked what's the start codon? Stop. What's the start codon? And I told you it codes for an amino acid. Yes, please. Yeah, it codes for met or methionine. It codes for methionine. And for bacteria, it's called FMET, formulated methionine. If you have a normal codon, supposed to give me valine, glutamic acid, I change one substituent, now it becomes what Bacola was saying. UGA, UAA, UAG. You go away, you are gone. Then I stop prematurely. That's called nonsense mutation. And then we have a silent mutation. And that's an interesting one we wish we all have. That's when you have a mutation that does not cause a problem. That's why, because the code is degenerate. What does degenerate mean? Degenerate means one amino acid can be transcribed by multiple different codons. So for example, valine could be coded by UGA, not UGA. UGA is a stop codon, I'm sorry. Like AAG, AUC, UAC. So you change one codon, but coincidentally, you change it into something that still happens to be one of the codons that are degenerate, and they code for the same one you were trying to have a target. That's why they call it silent. It's a mutation, but it doesn't have any problem. And you have the back mutation. That's when a mutated gene reverses to its original base composition. A mutation returns me back to my normal. That's what we wish for sickle cell anemia. That another mutation on top of them, they had a mutation that turned them from glutamic acid to valine. If they get an additional mutation that rolls them back into glutamic acid again, that's called back mutation. We love that. We wish that. But we don't know how to make it. If we make that, that'll be good. Frame shift mutation is when the reading frame of the mRNA is altered. So instead of starting from let's say I put multiple phone numbers. My phone number is 1, 2, 3, 4, 5. My friend is 6, 7, 8, 9, 10. My other friend is 11, 12, 13, 14, 15. Then I remove I remove one number, then the whole number is condensed. Now my number is 1, 2, 3, 4, 5, 6, 7. It went a number into my friend's number, and then everything got mixed up. That's called a frame shift mutation. Let's take a break. Take a break and come back in come back in
15.
let's come back in half hour. I think today by the time we finish lecture